Welcome everybody. I have the tremendous honor to do the introductions, very quick introductions, to welcome you here. I guess I'm going to say just three very quick things, make 3 points. One is, please stay after the formal presentation and questions, because there are some prizes to give out and some awards. So if you could just remain in your seat at the end of the presentation, that would be great. The second thing is to just give some context to the event today. This is just a tremendous event that has been going on for a number of years. And just in the last couple of years is co-sponsored and done jointly with the Brown Institute for Brain Science and the Department of Psychiatry, Human Behavior. It used to be the Annual Research Symposium on Mental Health Sciences. This event today is really being driven, and the energy behind this event keeps it going is really Steve Rasmussen, the chair of psychiatry. His tremendous energy and really due to his foresight that he realized how incredible it would be to engage the whole community, the whole brain science community, and that's what's been happening for the last few years. So I want to make sure to acknowledge that. And that there are a number of sponsors for this event today. A number of departments have contributed very generously to sponsoring the event, as well as the Norman Prince Neuroscience Institute. So I'm not going to say too much about our speaker, except that it's just really a great honor to have Jurgi Buzaki here. Chris Moore is going to do your formal introduction, so I'm not going to say too much, except that you're joining an incredible number of really inspirational speakers who are on the back of your programs if you want to look through. And finally, what I just wanted to say is I had the great honor actually of hearing Jurgi in Argentina. I had to go all the way to Argentina, and I could have just waited a few more months to hear you here. It was, I'd say, just incredibly inspiring talk that you gave. I was looking around the room when you were giving your presentation, and there were so many young people in the audience there, and they were just in awe of listening to you talk. And I read a little bit of the prelude to one of your textbook, and I was reading through, and you made the note that you never know when or where there's going to be this inspirational speaker that you hear in the audience, and you think, aha, that really changes the way that I'm thinking about my science. And I know for several of you here, that's going to be the case. I don't know that we know who you are, but for sure, a number of people in this audience will be changed by the end of today. Thank you.
Well, good afternoon. So without cliche, it's really special to have a chance to introduce Jurgi Buzaki, in large part because Jurgi is incredibly devoted to both logic and discovery. And you might say, well, all scientists are devoted to logic and discovery, and I might say, that's not true. Jurgi, you'd be remiss in introducing him if you didn't mention that he had won a lot of prizes, including the recent European Brain Prize, and that he has lots of papers, and he's somehow cited as one of the most cited people in some domain somewhere. And you notice the way in which I'm kind of waving my hands, because I think he has the same disrespect for prizes as we discussed this morning, and things like citation metrics that I do. Now, he does care deeply about a variety of things that are very important for understanding dynamics, including dancing, modern architecture. These are all things that consume Jurgi's complex brain. In all seriousness, that was meant to be a joke, but it didn't work. I mean, it's not a joke. He really is good at those things. I'm going completely off the rails here. So it's, in all seriousness, his deep commitment to logic has, without being facile in my summary of his trajectory, always led Jurgi to be obsessed with understanding in vivo systems and how they actually relate across multiple synapses and how they actually do so in behaving brains. Now, this is obviously in vogue. This is the Obama Brain Initiative, and this is the human brain mapping, one of the worst named things ever, but the big European Human Brain Mapping Initiative. Billions of dollars are being thrown at exactly sort of framing of the problem. But that was not the case at all in the mid and late 80s when Urey began having a whole series of just fantastic papers pushing that exact logic. Now, if you're going to have a commitment to that kind of logic, you also have to have a deep commitment to inventing the methods that allow you to actually go after the problem at the right level of inquiry. And so not only has Urey been focused on what I think he's contributed a great deal to why there is this renaissance at that level of analysis, and maybe that's obvious, but it's important to say, but he's also contributed a huge amount to the methods that actually allow us to record from enough neurons at once, analyze in vivo behaving systems, do the actual things that would allow you to get at those sorts of dynamics. You should read his book. Okay, I said that. Now, one thing that I really, I'm of a mixed mind about when introducing Urey, I've been a bit of a fanboy since 1991, when he is thinking about theta oscillations, because he was deeply thinking about how could dynamics in the brain code information in a way that would actually be packaged the right way, provided for me the most interesting and elegant reason to continue to think about long-term potentiation, which I was rapidly souring on as something I had to do every day in slice rigs. But that I think it was an 88 or 89 paper that I read, completely changed my view as to what would be possible in terms of thinking about dynamics. Now I'm of mixed mind about Urey because he's one of the world's best people to argue with, but I unfortunately find that everything he says makes way too much sense, and so I have to try to look for really good topics to argue with him about. Luckily this morning we started to find one based on whether or not discovery could be quantified as a rational metric across papers. I don't know if that's actually an argument. But the bottom line is he thinks incredibly logically and thoughtfully about topics and then lets the science follow after that. And that's something that, again, Diane was talking about ways in which talks can change you. I hope that's at least one thing that people take from this, is always to put the logic first and then invent the methods, invent the approaches after that. So it's an incredible pleasure to welcome you, Urey, to speak.
Thank you, Chris. Thank you, Diane. So after such an introduction, everything is just downhill from now on. So when I got this honor to be here, I got the invitation from Steve Rasmussen, who is a psychiatrist, and I thought I was going to talk to psychiatrists. And this is the reason why I have this extraordinary title here, because this is a very complex topic, very complicated things, but it doesn't mean that we have to give up just because something is very complex. So I'm trying to do my best, and I tell you something today that you don't have to take seriously. This is a progress report in my mind. So how do we think about these issues seriously? Let's take a simple organism. This is a very simplified, perhaps an imaginary organism that has to deal with the world. What it does, it generates an output. This is what brains do. They generate outputs, the result of which, for example, it can change a sensor. The sensor detects something that is fed back to the brain, and as a consequence of this, if it was good or bad, the brain takes notice of that. And you have noticed that I started with action rather than the other way around. Now, this hypothetical organism can work for one and only one reason, is because there are regularities and recurrences out there in the world. The most important thing for the brain to do is to predict the future. This little organism does nothing else, just predict the future based on the experienced past. Now, with evolution, things change a little bit, not fundamentally, that we add a loop to this input-output system, and another loop, and another loop, and another loop, and we keep adding many more loops. The goal of this entire complexity is nothing else, just predict the future. But in this case, predict the future in a more complex environment and at a much longer time scale. So for example, you come to graduate school, you invest 4 years because you predict that with a PhD, your life will be better. So this is good. I think all of us agrees with this. This is not cognition yet. At some moment, something interesting happens. The interesting thing is that these loops become very smart. They become so smart that they no longer need the world. They can look this is the point when the brain can start looking at its own computation. So it can try what-if scenarios even without moving a single muscle. And this is due to the act of learning and teaching. Now, of course, this learning can be phylogenetic, this can be ontogenetic, and I will show you examples for both. So my goal today is to show at least 3 examples that show this process that I call disengagement of the brain from the environment. Let's start with something simple. Every expecting mother knows that baby kicks are super duper important. The reason why they are, because the gynecologist will tell you that the number of kicks and the magnitude and so on correlates very perfectly with their Apgar score. But a psychologist would tell you that even your IQs correlate with how vigorously you kicked in the womb. But neither of them will tell you, why is it good for the brain? What does the brain benefit from all these kicks? Now, how do you study this? You can study it in various ways. The simplest thing is you go to the laboratory, you find the right species, which in this case is a ratlet or rat pups, and they do exactly the same, but they are prematurely born. When they are born, they correspond to about the transition between the second and third trimesters in humans. And you can see what they do is kick. Sometimes only the gastrocnemius is twitching, sometimes you've got a limb, sometimes you have a whole body startle reaction, and there are many more than just an isolated little human. So then you would like to know what happens in the brain at these times and why is this important. In fact, the question that we ask had nothing to do with this. We asked something different, namely we asked, what is the first organized pattern in the brain that occurs during ontogeny? And the rat is premature, so we had to go back from a little later all the way to P2, postnatal day 2, and see what patterns are there. And we know it from premature babies, as well as from other indirect studies, that what you get typically is nothing. There is no brain activity for a long period of time, just like shown here. And you look at multi-unit activity or local field potentials or movement, and then all of a sudden, out of nothing, there is a big activity. You zoom in, he said, whoa, this is beautiful. This is a spindle. So the first pattern that occurs in the brain is a rhythm. And many of you who have first, went through residency training in psychiatry and neuroscience and anything has to do with the brain, they say, oh, this looks like a sleep spindle. And in fact, it looks like a spindle. It's a very nice sleep spindle. The interesting thing is that they occur all the time. And they occur only when there is a movement in the body. So how do we know that? So for example, whenever we observe a pattern like this, we look at the body movement. They said, oh, every single time there's a kick somewhere, there is unit firing, and there is a spindle related to it. You can do a correlation, and you find indeed that this is an interesting pattern that is supposed to occur during sleep. But in fact, when you look at the forebrain of a newborn animal or a premature baby, you have no clue whatsoever from the forebrain whether the animal is in REM sleep, slow wave sleep, or waking. You can look at the hindbrain patterns such as respiration, cardiac cycles, and so on to classify the behavioral states. But here, every one of them is generated by some spindle. And this is interesting because at this early age, the IA spinothalamic afferents are already there. The thalamocortical fibers are already there at P1, P2. However, the long-range organization in the superficial layers of the cortex, they don't exist. So if this is the case, then there should be a perfect localization. That is, if you twitch this muscle of yours, then it has to be a representation, a topographic representation somewhere corresponding in your somatosensory cortex. And in fact, this is the case. This is in contrast, of course, with the sleep spindles, because sleep spindles are traveling. They occupy a very large amount of space, but they cannot do it here because there is no anatomical substrate for that. So indeed, again, you see a very nice topography here. So there is a pattern, a rhythm, that completely relies on input from the periphery. The spinal cord is the initiator. It generates a stochastic pattern that is a twitching of the movement, but from the twitches, the somatosensory information comes back, and this is responsible for this pattern. Now, to show that, you can cut the spinal cord. This is a spinal cord cut, And these are the number of events before the cut, and you can see that after the cut, they completely go down and almost completely disappear. Whereas above the cut, when the forebrain, this paw is twitching, then there is no change, of course, because we made a cut here. And of course, this is the same kind of pattern also in premature babies. Nothing, nothing, nothing. And with Rustem Kazipov and the pediatric neurologist was shown very nicely that every single time this anar saw pattern occurs here, or tracé discontinuo occurs, then you have a kick. Now, what does it do for you? Just think about the 650 or so muscles that we have in the body. You can put all these muscles individually, independently on a table. They are connected to the brain, and they start twitching. They don't do any useful thing to the brain because they are stimuli the way how experimenters stimulate the brain, randomly. However, these muscles are actually anchored to a skeleton. And once it occurs, then the degree of freedom among them is reduced tremendously. Those muscles that are agonists will work together, and they work against the antagonist. In other words, space issues are now translated into time in the brain. So every single time there's a twitch, it's a coordinated twitch in space, which translates into time, and by the time the long-range cortical connections come in, they have a guidance because of the timing and other type constant and so on to do this. In other words, how would the brain know a priori whether we are dealing with a body that has the shape of a snake or a ball or the way we look? In fact, you can ask, what happens if you give an extra limb? Let's say you have a fifth limb. Will be incorporated into the brain? The answer is yes. My old professor of anatomy, Seikei, he has done it in newts. It works like a dream. But what about giving you 2 hands or an entire new body? What will happen? This experiment we have never done. Unfortunately, this has been done by nature. And
twins, I think to our...
Tatiana and Krista Hogan are what's called craniopagus twin. Cochrane is the twins' pediatric neurosurgeon.
So these 2 brains are fused
amongst blood vessels,
they are
both
controlling
arteries
2 bodies
and veins, but they also have this connection
completely independently, but
what's called
not
the
so
thalamus,
independently, as you will see it in a second.
One in each.
They are very interconnected. They share a lot of things normal conjoined twins don't. They have the special abilities to see each other, see what each other's seeing through each other's eyes. Look at all that hair.
During this haircut, for example, watch Krista. Is she seeing through her sister's eyes?
Look at all this hair.
You think they do share?
Oh, I definitely do. I definitely do.
Felicia holds a pencil where Tatiana can't see it and asks her to grab it.
One in Mommy's hand.
So you got the picture. This is a very interesting experiment of nature. There is no amount of molecular biology, sorry for molecular biologists to say that that would give you this amount of learning. It has to be done on the fly. When you are 2 days old or 2 weeks old, the distance between your finger and your nose is different. So you have to update that distance with some update knowledge every single day. And these spindles, we think, are the culprit for all of this. What is interesting about this case, for the sidetrack for psychiatrists, is that even though one person can see what the other person sees, one person can feel what the other person touches, they are completely 2 separate individuals. They fight with each other, they do the same kind of things, even though it hurts when you hit the other one as well. So this is a very nice example to me for 2 reasons. One is because it's interesting and relevant. The second one is that this is a very good example that by the time, about the first week of life, when the long-range connections come in, the subcortical neurotransmitters come in, all of a sudden, this spindle becomes no longer dependent on the peripheral inputs. It becomes internalized, and it becomes a sleep spindle. But it doesn't completely break its relationship with the body. Tonight, when you go home and you go to bed, you will see every now and then you've got this huge body reaction. If you allow me to record from your brain, I will demonstrate that there is a spindle. So this seems that the early metastable state that will be stabilized by various speed later is preserved, but in a very small state window that happens before you fall asleep. So this was my number one. Number two, both of my examples are coming from the field that I work actively on, indirectly. This is the field of spatial navigation. For those of you who don't know not too much about spatial navigation, it works like this. You come to New York City, you have a map in your hand, you're coming out of the subway, and you have to find your way. The first thing you have to do is orient the map. If you fail to do the right orientation, you end up like me, that I'm going uptown 3 blocks before I realize I'm going in the wrong direction. So you have to orient that. The second thing is you have to get a relationship among objects in the environment, and it's called tessellation. Typically in a map, it's a square. But in a brain, it happens to be a triangle with 60 degrees, that is equilateral triangles. The third important moment is that you have to put yourself, your ego, into this map. Where are you related to everything else? There is another component that nobody talks about, is that you have to calibrate that map. There must be a mechanism in the brain that when you look at something, you said, oh, I have to walk only 50 feet, or I have to walk 50 miles. And unless you have the calibration signal, you cannot use that map. But let's start with the head direction systems, which are very important, because without head direction system, nothing else works. In fact, if you destroy the head direction cells, then there are no grids in the entorhinal cortex of the hippocampus. So where are these head direction cells, and what are they doing? How many of you remember Sherrington's writing reflex? Whoa, very few people. So Sherrington had a secret life, was working on regular things, but he always wanted to understand how come that you throw up a cat, it lands on its feet. Or when you put the animal or yourself on the side, you always right up. To make the long story short, he has done a lot of experiments. He blindfolded the animal, destroyed the vestibular system, cut the neck muscle, lots of things. And he concluded that things that are important for life are secured in a way that they have multiple representation in the brain somewhere. He never recorded anything from the brain, of course, but this was his conclusion. Head direction cells are like that. Head direction is the most basic and simple sense we have. Why am I saying that? Every other sense, all the Aristotelian senses that you know, they have complications. Vision has orientation selectivity, intensity, color, all sorts of things. So when you record from a neuron and it responds, you don't exactly know which of the 2 or 3 or 4 of the parameters are affecting the neuron. In head direction system, it's very simple. The head direction system cares about nothing else except which direction your head is looking at. I'm looking at the exit signal, and there are cells in my brain that fire about 50 to 80 hertz right now, and they will fire as long as I'm looking at that particular signal. Then I move around and move, and there will be another set of neurons while I make 360 degree of the head turn, and every single direction will be represented beautifully. So where is the information coming to this head direction system? There are neurons in the brainstem. They are mostly gravity related. There are neurons also, so the gravity related, just like in the writing reflex of Sherrington. Same thing in the supramammillary nucleus. There are many cells that are showing you some direction where your head is, but also where is your body and how it can be fooled by various tricks. But when you go to the anterior dorsal, the AD nucleus of the thalamus, it's a perfect system that cares about nothing else, just the horizontal direction of your head. Then you go to the prosubiculum, same thing. You go to the entorhinal cortex, layer 5, layer 3, same thing. You can go to the striatum, you can go to the parietal cortex, lateral septum, and millions of places, or many places in the brain. Therefore, it's, according to Sherrington, it's a fundamental thing to have. What is driving these neurons? Well, the wisdom is that it's the vestibular system, or vision, or any of the sensation. The reason why neurons in the brain fire, according to what I call Newtonian neuroscience, is because somebody drives them. And this is very clear in the visual field also. The reason why neurons fire is because somebody drives them. And here, there's a very simple scenario. Something has to be driving them. So how do you test this scenario? You have to recode from large number of neurons in the right spot. The AD nucleus is pretty small in the thalamus. So you have to do the right technology. Our specialty is that we don't recode with wires. We recode with these silicon probes that have high density, small damage, and you can go into a very small nucleus with 2 or 3 shanks also. So then the hypothesis that you're asking is this. The null hypothesis is that something such as vision or the skin receptors or head turn or something from the periphery is driving these neurons. And that's very simple to reject because you can eliminate all of these inputs by letting the animal fall asleep. So this is exactly what we asked. What happens when the animal falls asleep? So before I show you, you can see that there are simultaneous recordings in this is a mouse, in the AD nucleus, and the target, the direct monosynaptic target is the posterior subiculum. And there are this group of neurons that you can record from, and they have very beautiful tuning selectivity. I mean, this is incredibly attractive because the signal-to-noise ratio is just horrendous, very, very nice. So once you have recorded from a bunch of neurons, you can say, can I reconstruct their performance from their spiking activity? And this is what we have done. So for example, this is a group of AD neurons in the thalamus. So you can see there is some nice, interesting pattern here. And you can, at the same time, record with LEDs on the head of the animal which direction the animal is looking at. And the second step that we can do is that can we reconstruct this position from looking at the neuron activities? Same problem that many people at Brown have been struggling with, how to move a robot with the population vector of firing neurons. And you can see the reconstruction is pretty good because this is a very simple one-dimensional system. So what happens if the animal falls asleep, or in this case, it's the other way around. The animal was sleeping and it woke up. And you can see is that the activity is not random. It evolves very, very nicely. In fact, we can tell with high accuracy which direction the animal is looking at while in REM sleep. And then we go to slow wave sleep, which looks a little bit noisier, but when we zoom in, we also find that there is a nice evolution of the pattern. Except now the activity around this hypothetical ring goes 10 times faster. So what did we find so far? What we found so far is that when the animal moves the head around, the neurons that represent 50 degrees come after 40 degrees. And these neurons come after the neurons that represent 30 degrees. And it's not a miracle because the environment makes them fire one after the other. But it turns out when the animal falls asleep, the sequence remains the same. So it shows us or suggests that there is an attractor or there is a mechanism, there is an internally generated pattern in the brain that is mimicking whatever the animal has learned through phylogeny or ontogeny. So let me just elaborate on this a little bit. Let's take 2 neurons that are completely overlapping or mostly overlapping. Let's take that exit sign. Whenever I look at these 2 neurons are firing together in representing space, therefore together in time. No miracle that they have a positive correlation. Now I take 2 neurons that are representing the clock and the exit sign, and then I have to look at either here or there, therefore there is a negative correlation between the 2. So if there are neurons in this ring that are positively correlated and negatively correlated, there must be some neurons that are uncorrelated. So where is this transition occurring? Now, the interesting thing that you look at here is let me point out that this correlation occurs also during slow wave sleep and REM sleep, independent of the behavioral state, independent of the inputs from the environment. The relationship, the temporal relationship of the neurons remain put. So then the other thing is that you can see the calibration signal. This is 2, this is 20, this is 20. The ring starts moving faster during slow wave sleep, 10 times faster. And this is just showing the large population. You will see many of these figures. The hot colors represent either a correlation is increasing or the firing rate is increasing. And you can see that these neurons correlate positively with each other. These neurons correlate negatively with each other. and the transition happens at about 60 degrees. Now, we can look at it in a different way. Now it's a little bit more complicated because you can take pairs of neurons from the AD nucleus in the thalamus. You can take neurons from the posterior subiculum, or you can take one neuron from here, one neuron from there. And it doesn't matter how you play the game, and it doesn't matter which state you are looking at. Neurons that are positively correlated, there are neurons that are negatively correlated, and then there is a transition, and the transition always occurs at 60 degrees. So why is 60 degrees exciting? Because I showed you that the grid in the entorhinal cortex is the tessellation degree. Equilateral triangles have 60 degrees. And many people the Mozart group who discovered this, they are looking for mechanisms in the entorhinal cortex where the head directions, where the grid cells are, and they don't really find good mechanism. And later on, it turned out that there are grid cells on the parasubiculum, the perisubiculum, which are totally different anatomically organized structures, yet they have this same dynamic. So my pitch is that the head direction is given to them, probably from the head direction system, and that's the primary kind of organizer, and everything else is just a translation to that. Now, if these activities in the thalamus and everywhere else in the cortex and subcortical areas where the head direction cells exist, they preserve their relationship. What is their goal? What are they doing for the brain? And the answer is they can interpolate and they can extrapolate. So when I'm turning my head and something is blocking my view, or I close my eyes, things don't disappear. For an insect, for example, it's complete confusion. If you have seen an insect which is orienting towards the light, you put a shade there 5 billion times, the animal responds 5 billion times. It has no ability whatsoever to gap time in the hundreds of milliseconds or second range, whereas we are able to do that. So how much? So one way to measure this, we are looking at the population activity of these neurons here. Said, can we predict the timing of an arbitrarily chosen neuron downstream? And then we can ask, what is the time window of that prediction and the state of the brain? And what you can see, there are 3 different curves here, and then this is the bit value of the spikes. And you can see, yes, during waking, the system is most perfect. No doubt, no question about it. But what if you don't have the inputs from the periphery? Then the answer is, yes, you lose something. But what is left is the internally generated pattern, and it's enormous. It's 2/3 of the activity is generated for prediction very effectively in such a simple system as the head direction system of the brain. So here's another good example for me, that initially a system completely relies on the inputs from the brain, to the brain from the outside, and at some point is capable of disengaging itself and start generating internal patterns, and these internal patterns are put in good service of something like extrapolation and interpolation. And the sidetrack of this was, of course, that this organization is 60 degrees and so on. Now let's go on and skip the entorhinal cortical cells, because many people are working on it. Let's just go to John O'Keefe's place cells. So when the animal is moving from one place to another, place cells come into sequence. And the reason why they are coming into sequence, according to the teaching, is because they are driven by different constellations of the environment. So for example, this blue neuron, P1, is active because I'm moving towards the flowers. And when I pass the flowers, come to the flowers, they are firing maximally, and when I pass the flowers, then the activity goes down. This is what you can call, Hubel and Wiesel in the hippocampus. So this is one nice idea, and everything is driven from outside the hippocampus. The other way of thinking about it is that there is a self-organized system in the hippocampus that generates sequences. And I prefer that for a number of reasons. First of all, as you will see in the next slide soon, if the hippocampus, entorhinal cortex, was really a simple GPS, as you have read about it in New York Times and various places, then I wouldn't be able to give this talk to you. And the reason why I can give this talk to you is because I've got cell assemblies in my brain that are activated by some input, such as my introduction, or Chris's introduction to my talk, and then it will call up another assembly, which in turn will call up another assembly, and another assembly, and another assembly. And if you have time today, I can be here for the next 10 hours talking to you about whatever I want without any external input, because I have this ability in the brain. And of course, everybody knows that this is how it has to be. Finding it is a different thing. So now we are zooming into the hippocampus. If you read the literature about physiology and animals, this is the typical answer I get. The hippocampus is about navigation. The good news is that navigation comes in 2 different flavors in every single animal, at least in every single mammal. When you have a map, when you have landmarks, then you utilize the landmarks and follow them. But when you don't, you have to get back to what is called egocentric type of navigation. So what is it? It's called also dead reckoning type of navigation. Suppose that Chris brought me into this room, blindfolded. The lights are switched off. He leaves. I'm lost. But I can still have some home base. I can leave from my home base, turn around, remembering which direction I turned. I can go a little bit lateral, remember the angles, and come back to the same spot. And I can do this trick again from another direction and said, oh, this is familiar. I have been here already from 2 different intersections, and I have what graph theory says, a node. Once you have a node, then you've got some information about the layout of the environment. I can do the same thing with the flower pots or the chairs and so on. And then I can generate a map in my brain. When I go outside, I can draw a map. I discovered the world. I discovered this room with the help of my own actions. But then I can give it to you, and anybody who can read a map, because it's an allocentric information, anybody can use that information and navigate. So John O'Keefe suggested that place cells are doing this allocentric navigation. They are always relying on distant cues. Other people, like Bruce McNaughton, said, no, I think it's the local cues and the local optic flow and things like that are also important, and they may have something to do with this picture. Now, people who are coming from more cognitive background, they say, no, hippocampus is not about navigation. The hippocampus is all about memory. The good news is that memory of those things that we can consciously declare, they also come into 2 flavors. One is egocentric. These are our experiences. This is episodic memory. These are the most precious things we have. This is what makes us separate from everybody else, because all that knowledge, all that information is ours. So for example, when you discover something in the lab, we discussed it with students that everybody is entitled to make one discovery in his life. That moment will be yours. You will never forget that. You will remember when, where, and how, and what, and all the conditions. But if that finding is confirmed by my lab, and another lab, and another lab, and another lab, it's like when you are exploring that you've got different intersections to the same interesting finding. This way, you can strip off the spatiotemporal conditions of the fact, and the thing it just becomes a fact. And this is how we generate semantic information. This is a pointer, this is a computer, this is this, and this, and these are names of things and places and so on. So many people believe with me that indeed you have to discover the world in order to generate a map, and you have to have experiences to generate a general type of knowledge. So the past 30 years or so, based on physiology, the arrow was attempted to make from here to here. The people said, all we have to do is we have study place cells, and from the place cells, you give a little time or something like this, that you automatically have episodic memory. And it didn't work, and it cannot work for exactly that there is a dichotomy in both directions. So what I'd like to add to this is this. Nature initially invented a mechanism that allows to compute your whereabouts with the help of cues outside in the world. But then the system got smart enough, because we had many neurons, many loops, and the different structures to each other, that this pattern can internalize in such a way that now you can do a mental travel. And you can mentally travel back to the past or forward into the future. And this is exactly the same that uses the same substrate, it uses the same algorithms than navigation in the real world. The only difference is then when we are talking about memory, there are no outside cues. Most of the job is done to you by self-organization. So let's test this idea. The hippocampus is the right structure to look at because you need a large space to connect a lot of arbitrary information. If you are looking for some random connectivity in the brain, this is the CA3 region of the hippocampus, which is the best bet. What you see here is a single neuron filled here in vivo, and all the axon collaterals, which are the white lines here, are reconstructed in this three-dimensional structure. And you can see this is quite respectful. 450, almost half a meter of axons belong to this one single neuron. That corresponds to about 40,000 boutons. There are 200, 300,000 of these CA3 neurons in the rat that translates into 40 miles of axons and this many synapses. In incredible, dense, recurrent connectivity. 80%, Rebecca, maybe the number is right, 85% of the connectivity in the hippocampus is, or the axons and everything is coming from within the structure. Only a minority is coming from the outside. So this structure the goal is to connect everything with everything else. So let's see what is it good for. So if I put this one neuron into the right perspective, then one neuron can reach to 1/2 of the structure, or sometimes 2/3 of the structure. So this is an interesting organ which grows differently than the neocortex. This is a single giant cortical module. It just grows, and the goal is that no matter how big the size is, everything can be reached, every neuron can be reached through a few steps. So this is the point I'd like to make now, is that if there are 200,000 CA3 neurons, and you want to make sure that you can go from anywhere to anywhere else, all you would need is one neuron is connected to 16 other neurons. 16. But then you have to go from one neuron to the next neuron and next neuron, very, very long chains in order to deliver a message or to find a neuron that is relevant to generate an output. And von Neumann suggested that those systems that you have to go through multiple steps, and many steps, they have very low logical depth. Systems that spit out the results after 1 or 2 steps of computation, very, very high logical steps, this is the hippocampus. So instead of 16, now we've got 20,000 to 40,000 possible connections from any neuron to any other neuron. So now you can go from any neuron to visit any of the 10 billion synapses in 2 steps. And these 2 steps take only 100 milliseconds, which is the cycle time of the hippocampal theta oscillation. So now we have a substrate. We have a tool, the electrode. We would like to test the hypothesis, what I was just talking about. The next slide summarizes 30 years of very hard work of the best people in this field. What it shows you is an action potential of a single neuron in the CA1 region of the hippocampus, whose firing activity can be translated into a receptive field, if you want, or the place map, or place cell map here, and you can see what happens here. But if you listen a little bit more carefully to this neuron, you will hear that it fires rhythmically. So on this trial, this neuron fired at 8.7 hertz. Now, there are many of these cells that surround the electrode that we are putting into the hippocampus, whose membranes are also fluctuating, and their membrane potential is generating extracellular currents or extracellular voltage that you can measure outside, and this is the local field potential. And that local field potential change happens to be 8 hertz. So now we've got 2 oscillators. One is a single neuron, the other one is all the other neurons. It's a very interesting thing if you are asking yourself that these oscillations that are 8 hertz are made out of a myriad of oscillators whose frequency is higher than the common rhythm, and you can figure out why if you want. But the point here is that you've got a oscillator here, an oscillator there. They have a non-integer relationship between the two. The result of which is interference. And interference is good because it gives you incredibly nice precision. And this is another nice discovery by O'Keefe. called phase precession. So then the animal enters the field, the neuron fires on the peak of the theta cycle, the animal moves into the middle of the field, there are more action potentials, the spike side, the trough, and the animal keeps moving or moving out of the place field, the phase keep processing backwards. So his argument was that, this is a further proof that you have to take only a snapshot of about 100 milliseconds, looking at the relationship between the spike and the theta phase, and you will know where the animal is. This was the point when I became very much interested in, because now time entered into the place field literature. And I said, well, this is pretty interesting. And in fact, this is O'Keefe's beautiful demonstration that if you look at the relationship where the animal is now in the track, and then you look at the phase, then there is a systematic relationship. Now we can look at two neurons. When two neurons are together, such as the green and the red neuron, and now the animal is running in this direction, you're asking, what is the relationship between distance and time? So the distance between the two representations of these two neurons is about 80 centimeters, and then you can ask, when the neurons fire together, what happens to their temporal offset? And the blue thing here shows the speed of the animal. The longer the blue line, the faster the animal runs. And you can see sometimes it's long, sometimes it's shorter. Independent of the animal speed, the time here is always about the same time. In other words, here's a nice demonstration that there is a distance representation by two neurons. These neurons can fire together within the theta cycles. And when they fire together the theta cycle, they've got a fixed relationship. And this fixed relationship doesn't really change very much. In fact, you can do another experiment that I learned from both O'Keefe and Bruce McNaughton. If you put an animal into a larger arena, then the place fields move away from each other, or vice versa. A smaller arena, place fields move towards each other. So in this case, this is two neurons, 1 meter long track, and this is 2 meter long track, and they move apart. And you can ask the question, what happens to their timing relationship? And the answer is pretty simple. Nothing. So what it tells us is that there is a hierarchy, there is an organization in the brain that has to respect time. Whatever you is a consequence of these rigid time relationships. Another way of translating this, that the world or the present or perceiving the world will suffer because of these time constraints, what happens within a single theta cycle. So if you put an animal into a small room, then the place cells from each other are very small. The resolution of the environment is very large, but the temporal offset is the same. You put the animal now into a football-sized field, the temporal relationship will not change. The consequence of which is that the resolution of the large environment will be very poor. It has a further consequence, but I will go to the next slide before I do that. So now we learned that there is a very nice relationship between distance out in the physical world that you can measure with a ruler, and timing within the theta cycle that you can measure with other various tricks. So let's just do this. We already learned that 80 centimeter distance in the dorsal hippocampus is represented by a time offset of 80 milliseconds. Now, if you have two place cells that are 100 centimeters from each other, their temporal offset is 100 milliseconds. In the third scenario, the distance between the two representations is 150 milliseconds. What will be the time offset? If you don't tell me, I'm not going to move forward. Does anybody dare to say anything? So it was 80, 80, 100, 100, 150, and? Thank you for saying that. Of course it's not. And the reason why it cannot be, because the theta cycle is only 130 milliseconds. And indeed, this was our guess. When you have few data, as we had with George Dragoy, initially, we had only 400 pairs. And then we were here on the slope, and we said there's a linear relationship. But when you have 27,000 pairs of correlations, then you can see that, oh, it just happened that it was linear, but it's part of this sigmoid. And the sigmoid is due to the fact that you can put in minimum number of information only to this theta cycle because of the time constraints. So what is the translation of this? The dorsal hippocampus of the rat can see the environment in its high resolution for about, let's say, a meter or so. In order to resolve the next part of the world, you have to move forward. And that's an interesting thing and good news, because this is exactly what happens with our memory recall. We have extraordinary amount of memory somewhere stored, but only snippets come up to the level of consciousness. And I can give you a good recollection about the previous 2 or 3 slides and what's coming next and so on, 7 plus minus 2, but I have no clue what was before and what was going by the end of the talk, because they are here at the level of this sigmoid. So that indeed, there is, wherever I look, wherever we go, there is perfect parallel always between what problems in memory occurred and problems in navigation occurred. So now we are going to the true test. What is the true test? First of all, the only way how we can judge that the theory is good if the theory gives you good predictions. O'Keeffe's theory is wonderful because it gives you perfect prediction. What are the predictions? If you imagine that somehow we freeze a rat in space at one point of the environment, then the prediction is that there will be always a subset of neurons somewhere in the hippocampus that will determine and fire, because they will determine the xy coordinate of Cartesian space, and this subgroup of neurons will fire forever, as long as you can maintain the theta dynamic by some trick. Those neurons that don't fire, they belong to representation in another phase. From those neurons in this small subset that happen to fire, they fire always in the same phase of theta. Because if they would move, according to what I've shown about phase precession, it means that the animal is translocating in the environment. So we are testing this. I'm just testing 2, but I'm showing you the evidence only for 1. So what we are expecting, if the animal is not going anywhere, then only a subset of neurons should be able to fire, and they do it forever. And of course, this is not what happens. So what we do is use a hippocampal-dependent task, which is very simple. The animal has to go to the right, collect water report, and then has to remember that, oh, I came from the right, next time I have to go to the left. It's a spontaneous alternation task. It's hippocampus-dependent, especially if there is a delay here. So between the choices, we ask the animal gently over several days of training, please always face that direction, same direction, and run at about the same speed. So in our power, we have done everything possible to keep the environment constant, environmental inputs constant, and keep the idiothetic information from the body constant. And now we are testing what happens, and then this is what happens. So now this is not distance, but this is time of the time the animal runs in the wheel, 15 seconds. And you can see that on every single trial, this neuron likes to fire for a short period of time, for about 1 second or so, boom. This one is later, and there are many neurons. You can collapse these neurons into a session average. And if you have enough neurons, then you can see that the entire journey, the entire trajectory of the animal can be reconstructed from these neurons. Now, there's something remarkable about this, that the lifetime of these neurons, that is, how long they are active, is exactly the same as if the animal were navigating in a real environment. So I can give you this data set to anybody who is an expert in the place field literature, and he can't tell whether an animal is not going anywhere because it's just running on a wheel or traversing the environment and translocating. The second important interesting thing about this is that there is not a single neuron that would persistently fire. That would be the prediction of the O'Keeffe theory. Instead, they have this nice sequence. Now, the good news about this sequence is that somewhere in this trajectory, the virtual trajectory is nothing else but the packet in this CA3 region that I showed you of the activity goes in a tortuous manner from one place to another in time. And that trajectory is useful because that contains the information about the past and the future. So now we are trying to dig out that information. I show you first one neuron. So the animal is in this wheel. There is a T base here. You heard the firing of the neurons. The animal went to the left. Just complete silence. Goes to the right. Okay, so we have a hypothesis. Now it's firing. We are testing the hypothesis. Oh, in the first observation was that the animal went to the left. Same thing. We confirmed the hypothesis. And the animal comes back. Said, oh, no firing. The animal has to go to the right. We had the hypothesis. We confirmed the hypothesis. Now you can publish a paper. Except when you start looking at it more carefully, said, okay, this is firing now. Then we already have a judgment. I said, oh, it was firing. The animal has to go to the left. Everything, correct trials, correct prediction, everything is fine. Now, no firing. Remember, we can look at the animal. I said, we can make a prediction based on the animal's behavior. And the prediction from the animal's behavior would be, the animal has to go to the right. I'm looking at the neurons, or the neuron. The prediction is always also that the animal has to go to the right. But sometimes conflicts occur. And the conflicts occur right now because the neurons started to fire. Another couple of spikes. And then you can hear there will be several more spikes. Spike, 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 spike after that. Now we're hesitating whether we should believe the neuron or if we should believe the Bayesian prediction from the animal's behavior. And then you can see that the neuron tells us which direction it really has to go because the neuron somehow contributed to the decision the animal made. Now this is a good trial. It was completely silent. The animal will go to the right side. So remember, that was an error from the animal. And now it's interesting that we know that the animal has to go to the left. And it doesn't. It goes to the right. It comes back. The neuron says, oh, you made a mistake. Turn to the left. And then the animal will turn to the left. So this is an incredibly nice neuron. It shows the difference between, left and right. But it's completely useless in guiding navigation or guiding motor reaction of the animal. Why? It's because this neuron fired shortly at the beginning of the journey. And there is another 14 seconds or so that will elapse, and the information should be kept somewhere. And the good news is, there is another neuron, another neuron that are differentially involved. And then you can set up or list all your neurons and ask, if I know the information, whether the animal will go to the left or will go to the right, and I make this trajectory artificially, by lining up these neurons in a line, such as this one. This is a trajectory. This is the activity packet is traveling in the three-dimensional space of the hippocampus. How these neurons, where the identity here and here are the same, differ when the animal goes into the other direction. And what you see here is that no statistics are needed. You can take any slice of time, short or long. You can make a very intelligent guess whether the animal will go to the right or will go to the left, with 90% accuracy, if you have enough neurons, including errors, 15 seconds down the future. So 15 seconds ahead, you can make a good prediction in this constrained, simple environment, whether the animal will make an error or not and which direction it will go. So we can do, after this experiment, one thing that we like to do best in the laboratory, which is rejecting somebody else's hypothesis. Say, the hippocampus cannot help. Every single time it has an initial state, it will generate sequences forever. This is its job. But it doesn't discredit O'Keefe at all, because every single time when there is an external input, this trajectory can move. For example, if you would ask me a question, then you would break the flow of my thought, and my trajectory in the hippocampus will jump to another place. And the more questions you ask, the more the trajectory will be affected. And if there are more and more significant inputs from the environment, then the hippocampus can follow that trajectory that is imposed upon. In other words, there are two ways to do this. Is it possible that I've been talking for 2 hours now? Because I have the wrong timing here. OK. So this is all good. You present the evidence. You are very happy. But then the problem are always with psychologists. They come back and said, there's not a proof for episodic memory. I understand that there are sequences and so on, but episodic memory, according to Tulving, by definition, is in the recall. So unless you show us that the animal recalls the same sequence as the deduction of the sequence during encoding, we don't believe you. So of course, we can't do this experiment because it's difficult to communicate with rats and mice. But I was at Hebrew University at the time when we were doing this experiment, and Itzhak Fried was there, and I said, well, this is an interesting problem. He said, well, we are just recording from humans, from the hippocampus, perhaps, from the entorhinal cortex, perhaps. We have few neurons, but we can ask many, many questions from them. I said, how are you asking the questions? I said, well, in the form of a movie. I said, this is fantastic, and this is the companion paper of our paper, that this is what they find. What you will see is a single, perhaps CA1 neuron in the human hippocampus after showing clips of movies.
He's been filmed smoking marijuana.
Powerful spiritual significance to the native peoples. Niagara Falls.
You are gone! The famous couch jump.
So you can hear the firing of the neurons, you can see the rate of firing.
The power of the force.
Don't try to fragment.
So after 2 trials, you can see that there is, whenever Tom Cruise comes back, then the neuron goes bizarre.
You are gone! The famous couch jump.
So this goes on and on and on, but is not anything more than what I have told you and convinced you from rat experiments. But the reason why I'm showing you this movie is because
the famous couch jump.
At the end of the experiment, after the exposure, they can do something that I cannot. They ask the person, what did you see? And now you can hear the utterances of the subject that tells you very nicely whatever he remembers, and the recall in this case was perfect. And since you are an intelligent audience, you can predict what's going to happen here. But listen, that first the neuron fires, and then there will be an utterance. That activity goes in the opposite direction. During exposure, it comes from the environment to the hippocampus. Now it came from the hippocampus and outside. So just to repeat and summarize what I had said so far, there is enormous temporal constraint in the brain. Everything that we see and do and is a consequence of this temporal constraint. We cannot put a lot more information in the hippocampal theta cycle than it is allowed by its internally generated pattern. But I also would like to emphasize that this third experiment, very beautifully, as far as I'm concerned, showed that initially the brain was set up for generating sequences with the help of external cues during spatial navigation. And then at some point is capable of disengaging from the environment and use the same algorithm to generate internally patterns that allow the brain to go forward and backward in time without any cues from the environment. So I can skip a little bit with this. I'm running out of time, but what I'd like to say that this sequencer localization is very interesting. What 2 things we wanted to know about it is that how is it organized? And what is the lifetime of the neurons? Why is that this cell lasts for about 1 second, which is about 7 theta cycles? I skip the latter one, but I still like to show you that this is a very important pattern here. What is the mechanism behind of these phase offsets? And I give away the clue right away. Chris can guess. It has to do with inhibition. So what we do is either silence perisomatic interneurons or silence interneurons that are targeting the dendrites of the pyramidal cells. And I skip the nice experiments. I just come to the conclusion. Oops. Even though these are nice experiments, I just don't want to keep you here forever. So under normal condition, this is the theta cycle shown. Twice you can see that every single phase of the theta cycle is exploited for representing information. Now, this is good news for those of you who believe that oscillations are good for something. You may remember that when we started this, the criticism was this. every single time there is something interesting happens in your brain, there is no oscillation. Every single time when there is sleep or something bad happens, such as epilepsy or other things, then there is enormous synchrony. And that, of course, is not good because now the bit value of information from spikes is decreasing because many neurons are doing the same thing. Now, it turns out, and Bill Newsome, for example, and Shadlon, they had beautiful experiments and showed that independence of single neurons in V1 is largest when the animal is attending a target. Whereas the argument was in one of the papers is that when oscillation happens, everybody fires together. And this is not true. Hippocampal theta is doing something very interesting. Is it not synchronizing? It is not desynchronizes, but something in between, what I call metachrony, which is that there is a beautiful sequence representation here. Every phase of the oscillation is representing something else. The bottom of the theta says, you are here. And those neurons that representing the here now are firing maximally. But that knowledge is helped a little bit by those cells that used to be most active in the previous theta cycle on the ascending phase of the theta, which would be here in this part. Now, those neurons that will be most active in the future are helping out a little bit because they have got this long shoulders or tails, and they are helping out with this. So the here and now is nothing else but a continuous evolution from the past to the future. If you block perisomatic inhibition, in this case, we just blocked about 15 to 20 potentially perisomatic basket neurons, then we will find that this relationship breaks down completely. And instead, those neurons that fired early, they fired the trough. Those neurons that fired late, they also fired at the trough. And I assume, which we didn't do, if we will be able to block all basket cells and all inhibition, all these neurons would fire completely synchrony in a few milliseconds, and then you would call it an interictal spike. So in speculation or interpretation, the downstream readers say, aha, there is a sequence, that's sense, that means something. But when you deliver something like this, it's not readable. There is no information contact. And the reason why I dare to say that, we have done another set of experiments with cannabinoids, and we showed that this is exactly what happens when you give cannabinoid agonists. These are the CB1 receptor agonists, that this is what happens. The temporal resolution, the temporal relationship at the tens of millisecond scale is impaired tremendously, but place cells remain intact. So while we have a hippocampus where the place map is there the same way as before, the only thing that is changed a little bit is the relationship of timing between neuron 1 and neuron 2. And the same animal which has a perfect map cannot solve a spatial task. Presumably, because there are downstream readers, cannot make a difference in their proper time resolution, which is the AMPA time scale. Now, let me return to the big picture that I wanted to convey. Remember, doesn't matter how small or how large an organism, its goal is to predict the future. Simple organisms can predict the future in the tens and hundreds of millisecond time scale in very simple environment with very few variables. The many, many loops that we add, they help us to extrapolate and interpolate. But the big claim of the talk is that there is a moment in evolution, or perhaps ontogeny, when you disengage from the environment, and this is the moment when you try to do what-if scenarios without actually acting out. You just evaluate them mentally, whether I should stay in graduate school or quit and go to medical school or Wall Street or somewhere else, and then without moving any single muscle. Thank you very much.
I have a question. So do you think time cells, hippocampal time cells, are they something new, or are they just another manifestation of what you're talking about?
This would be a fair question if Howard Eichenbaum was here. So remember, O'Keefe said there are place cells in the hippocampus, and place is number one. And because his theory is good, he makes an explicit prediction. Explicit prediction is that out of every 200,000 pyramidal cell will be a place cell somewhere in the dean's office or in wherever you take the animal, but somewhere. Now, as I showed you, that we can measure the sequences, and that sequences evolve in time. And we can predict very precisely where the animal is in time from the beginning of the trial or from the beginning of the place field. So for convenience, we can call it a time cell. And this is what Eichenbaum does for a good reason, because as time is equivalent information, plus he's teasing the place field people that said, how do you dare to call these place cells? And I say, I'm ignorant. I just know that the hippocampus is a dumb structure. All it does is compute something that responds to the inputs from the codex. Whatever it is the computation. Whether we call it time cells or place cells, I'm not saying it in public, but it doesn't make a lot of sense to me, because the two are connected through speed. They are linearly connected. It doesn't matter which one you say, distance or time and speed. So, and you can try whatever you want. One gives you the other. So it's a good way of talking about time cells, but I think it's exactly the same thing. The good point about Howard Eichenbaum is showing that he does all these experiments when place is excluded, because the animal is running either in a wheel or, in his case, on a running track. So he can say these are not about space only, but it's not about time either. Time happens to follow every single thing that happens in the universe, because there is evolving, computation happens over sequential activation. I could have given this talk without mentioning time or space, and I would have the exact conclusions.
Hi. I wonder if CA1 anatomy kind of place any constraints on how these nodes are organized. So my expectation is that the activity, when it travels from one node to the other one to the other one, should go through adjacent nodes.
So that's another very interesting question. Most of the things that I recorded from was CA1 region, but we believe that the activity actually happens in this n-dimensional region of the CA3 recurrent collateral system. Now, activity travels from the dorsal hippocampus to the ventral hippocampus, and it takes about 70 milliseconds or 180 degrees. That is half a theta cycle. So that's an incredibly interesting question, whether information is segmented in time and space, in brain volume, or somehow some things have to be together and read together in an amper time constant. How is it possible that the ventral hippocampus says the same thing as the dorsal hippocampus if the activity is deliberately delayed 1 NMDA time constant? Now remember that this sausage, the hippocampus, is connected to the neocortex in a nice indirect topographical manner. So even going back to Rebecca's question, is that the thing that we are usually not recording from, but we can, is the ventral tip of the rodent hippocampus. This is the area which is connected to the prefrontal cortex and the amygdala and other things. And when the neocortex grows, it becomes very large, and it's filled up with association areas. This tiny, 1/5 of the hippocampus or 1/4 of the hippocampus starts to grow. And this becomes the uncus and the body of the primate hippocampus. And the rest of the hippocampus that we are recording from is only the tail. So this connectivity is there. That's why it is very important to know, are the downstream readers have segments to read out their information? And what are those segments? 1 millisecond, 5 millisecond, or 100 millisecond? And there is no good answer for this. Nobody is really looking at whether the information is read out over time. I know that it happens. Sequences are very important. Any brain can tell the difference between ta-da-da-da, ta-da-da-da. So this sequence difference is somehow coded in the brain and could be read out, but there is no experiment. There's none that would address that issue. Can you say it again? So there is phase preference, but phase preference is temporal. The essence of phase precession is that depending where the animal is, the phase can vary. So individual neurons, when you record them over time and average them the way how we shouldn't do, but we do, is always have a preference to the trough, every one of them. But if you do a snapshot and record all of the neurons at once, then you will see that these neurons fire 3 action potentials in the trough. These neurons only 1 or 2 at the ascending or descending phase, and others, and many, many neurons. The neurons are in different phases. And when you can repeat the same behavior, so then with the same exact spot, then this sequence is exactly the same. No, not at all. So that has been addressed by O'Keefe very early on, whether there is a topography in the hippocampus that corresponds to the topography of the environment, and it's not. So neighboring neurons have the same exact probability of representing the same amount of space or different amounts of space, or different parts of space. And that's the answer. Yes? No? No more questions? Okay, thank you very much.
The most important thanks, I just want to be sure to acknowledge the people who did the work for organizing today. Holly and Fatima, we owe you great thanks for all of your energy and hard work to put on the event today, with assistance from Tracy, Debbie, and Sherry. So thank you all very much.
So you're first.
Oh, I'm first. Okay, so these are quick announcements of poster awards. It was an incredible showing. What was the total number of posters? 140? 150, yeah. Amazing. So let's see. First place, this is the undergraduate student award winners. Adriel Barrios-Anderson, inter-alpha inhibitor proteins reduce the quantity of microglia in the hippocampus after hypoxic ischemia brain injury in the neonate. Is that presenter here or not? Nope. Okay, we'll make sure they get the prize. The next graduate student, medical student award winners, in second place, let's see if they're here, Tracy Sutter or Suter. Oh, great, excellent, come on up. And as you're walking up, I'm going to mention also first place winner, Hei Yang-Xing.
Just to say that the first place award winners is a $100 gift certificate, and the second place $50.
Should have said that. Big deal. Congratulations. Okay. Are you next?
I think I'm next.
Great.
Okay, so now we'd like to award the Research Assistant Award winners. And the second place award winner is Lacey Craker, if Lacey is here. And the first place award winner, Chloe Serapis, if Chloe is here. And moving quickly to the Clinical Psychology Resident Award winners, our second place award winner is Lily Brown, if Lily is there. And our first place winner, Joshua Kemp. Okay, and now moving to our postdoctoral clinical department award winners. Second place is Leslie Ann Brick. And our first place award winner, Carolyn McCormick. Okay, now we're going to switch.
Postdoctoral Basic Science Award winners, An Min Kyu, and in first place, Shane Crandall. Shane is here. Congrats.
And finally, our resident award winners. Our second place award winner is Catherine Rideout. And our first place award winner, Robbie Fenster. So we'd like to thank all of you for coming and participating today in our Mind Brain Day. It's a day where we hopefully have had a chance to interact with each other and learn from each other. And once again, I want to thank Dr. Buzaki for his time and his expertise and his lecture today, which hopefully we're all going to go back and try to figure out some of what he said, and also be able to kind of integrate it with our own work. And I know that his work has been seminal for our work. So thank you again.